Good afternoon, everyone, and thanks for attending this liver seminar. As you know, we are starting the 5th edition of this program, and today we have the pleasure to have live the invited speaker here in Barcelona. So we are truly happy. Today we have the pleasure to have Meritxell Huch with us. I don't think she needs a presentation, but I will give some brief remarks. So she obtained the doctorate at the Center of Genomic Regulation here in Barcelona in 2008, until the guidance of Cristina Fillat, who is nowadays a principal investigator here in IDIBABS. Afterwards, she pursued a postdoctoral fellow at the Hans Clevers Lab in Utrecht, in Netherlands, and this was a very productive indeed postdoctoral fellowship of more than 6 years, more or less, describing key methods to obtain stable stem cells and also to develop organoids, and all of this nowadays I think is worldwide used as a tool for the research in liver disease. Afterwards, she moved to Cambridge to become a principal investigator, but more recently, 2 years ago, she decided to move to Dresden, becoming a group leader at the Max Planck Institute of Molecular and Cellular Biology and Genetics. As you very well know, she published more than 60 papers in very high ranked journals, including Nature Cell Biology, Cell Stem Cell, Nature Medicine, and so on. So indeed, it's a pleasure to have her. And as you know, her lab is mainly focused on unveiling the biological processes that control adult tissue homeostasis, tissue, liver, pancreas repair, and especially their deregulation in liver diseases. So for that, it's my pleasure to welcome Meritxell to the liver seminars, to IDIBABS and Hospital Clinic of Barcelona. And today, her talk is entitled Liver Organoids for the Study of Regeneration and Cancer. Thank you, Meritxell.
Well, thanks, Jordi, for the beautiful introduction, and thanks everyone for coming and those that are online as well. Thanks for the invitation to be here at IDIBABS. I had only been maybe once when Cristina just moved here, but I was not a PI, I was a postdoc back then. So yeah, it's my pleasure to be here today. I'll be using the laser pointer so then everyone can follow. And as Jordi mentioned, my lab has worked extensively on organoids, liver, pancreas. We started with stomach. This is the outline of my talk. I'm going to give you a little introduction on organoid field in general. Then I will be focusing on liver organoids, of course, how we've used them to understand some questions on liver regeneration, in particularly epigenetic regulation and cellular niche. And I'll be finalizing with modeling human liver disease, in particularly cancer, and how we're using this to study disease mechanism. I'm just going to do a bit like this, maybe better, yeah. So basically, I like that slide to kind of summarize and give a perspective of the organoid field. And actually, the word organoid has been extensively used lately, probably in the last 5-6 years, but actually, the first time that the word organoid was used was in 1946. And that was a work of Cochrane and Smith. It was published in American Journal of Canadian American Journal. And actually, what they were using that word not to name what we now call organoid. That's why I liked them to call them pre-organoid era. But they were using it to label structures that they had in culture that were actually teratomas. So they were using the word to label something called cystic organoid teratoma. Actually, there was a teratoma was grown on the genital ridge of a newborn. They put it in culture and they used for the first time that word. This has been actually afterwards used to label some culture structures, three-dimensional structures that other groups, including Michaelopoulos, which of you might know for his huge contribution on the liver field. So actually, I could say Michaelopoulos had the first paper that says hepatocyte organoids. But still, it's before what we call the modern organoid era. What we were having up to now were just cells that people tried to put in culture and try to see whether they could recapitulate something of the tissue. The breakthrough started with Yoshiki Sasai in 2008, and in 2009, in parallel, I would say, the work of Hans Clevers with Toshi Sato with intestinal organoids. I will not be talking about Yoshiki Sasai's work on how his lab developed. They even didn't call them organoids at that stage. It was just pluripotent stem cells, trying to derive them into cortical tissue, and actually they did realize that they were recapitulating part of the architecture of the tissue ex vivo. In 2009, actually, my colleague, Toshiro Sato, in Hans Clevers' lab, he developed the first adult tissue-derived organoids. So in the upper part of that slide, you have now a summary of the different organoid cultures have been developed directly from the tissue. In the bottom part of the slide is the different organoid cultures have been developed from pluripotent stem cells. Why we call them now the modern organoid era is because for now, to call something an organoid, a culture that we grow as an organoid, it has to fulfill a definition whereby it has to be a three-dimensional structure that is formed by self-organization of the cells. If you paid attention to the video, you would see that the cells were dividing and making a bigger structure. It will be derived either from pluripotent stem cells, fetal or adult tissue, either stem cells or even differentiated cells, as we're going to see for the liver. They have to retain the capacity to differentiate into different cell types, or at least some of the cell types of the tissue. And finally, they have to be able to recapitulate at least some of the function. And using this criteria, several tissues, adult tissues, now I'm just focusing on adult tissues, have been used and to generate organoid structures from different organs. As I said, everything started from the observation that intestinal stem cells self-organized in vitro and could generate three-dimensional structures that would retain some of the epithelial cell types as well as the function of the intestinal epithelium. We followed up with the stomach organoids, and of course today I'm going to focus on the liver ones. I don't think I have to tell you this, so I'm going to go very, very quickly, just to remind you on one aspect of the liver, and it's how the liver develops. So we have several cell types in the liver tissue. You know that probably much better than myself. We have hepatocytes and ductal cells, cholangiocytes, I'm going to call them probably ductal cells along the top. These ones are epithelial and come from the endoderm tissue during development. And as you very well know, the tissue has a beautiful architecture whereby these hepatocytes are sandwiched between endothelial cells because, of course, the liver is not only about the epithelia, but there is also a non-epithelial counterpart of the tissue, endothelium, but also mesenchymal cells, immune cells, and they all work together to make this beautiful architecture. Different organoid cultures have been developed from the liver tissue, or trying to mimic, or they have been used the word liver organoids. And here I'm just showing you the work, and this is the only slide on that, from Takanori Takebe, and where he was the first one to generate liver organoids from pluripotent stem cells. That requires that these pluripotent stem cells, they have to be differentiated into the endoderm, then specified into hepatic epithelium, then differentiated into a liver bud, and then they put them in three-dimensional structure, they make these liver bud organoids. Then now they put on top, if you want to include endothelium, you have to then take again pluripotent stem cells, differentiated them into mesoderm, differentiated them into endothelium, and then they start to mix these populations. The group of Ludovic-Vallier did similar things, trying to, instead of making hepatocytes, making cholangiocytes. These cultures, they retain some aspects of the liver development, but they are still embryonic like epithelium. And this is not what my lab is working on. In my lab, we've always worked, and when I was a postdoc and afterwards when I had my own lab, we've always worked starting with the tissue. And this slide tries to summarize the different liver organoids that have been developed from the tissue, from the liver tissue directly. And very recently, in a consortium consensus paper that was published this year, we redefined how we've been calling them. This is the first organoids I developed from the liver. Back then, we didn't know the cell of origin, so we called them liver organoids. Now they've been reclassified as cholangiocyte-derived organoids, and basically the liver organoids for the different types of liver tissue have been classified according to the cell of origin. So now we have cholangiocyte organoids, which is what I'm going to be talking mostly today. But we also have hepatoblast organoids, so taking from the hepatoblast, the embryonic liver cell, and generating hepatoblast organoids, as we call them now. These cells, as you will see next slides, I'm not sure whether I don't think I have that here, otherwise it would be no time to show you what I wanted to show. But we showed in my lab that you can take very early on just a specified hepatoblast in 10.5 from the mouse. You can put it in culture, and they can generate now both hepatocyte-like organoids and cholangiocyte-like organoids. I'll be happy to go into questions if you have some during the talk. Also, the group of Hans Clevers and Roel Nusse in parallel in a back-to-back paper, they showed that you can take hepatocyte cells from the mouse adult liver tissue or fetal human liver cells and put them in culture, and they can generate these structures that they call hepatocyte organoids. And as I said, I'm going to be focusing on what in 2013 we called liver organoids that now have been word as cholangiocyte-derived organoids, and how we've been using them to understand mechanisms of regeneration, focusing on epigenetic regulation and cellular niche. And this is what I'm going to be focusing in the most part of my talk. How is liver regeneration at the cellular level? You also know that very well. So the liver has this huge capacity to regenerate upon toxic injury. I'm talking about toxic injury. And the way it does it at the cellular level, and this has been studied by many groups, including us, but many, many others, is when during homeostasis, as well as during liver resection or acute injury, as far as there is preservation of the healthy cells, the epithelial cells, which are the damaged ones, are going to self-replicate, and hepatocytes are going to make hepatocytes, ductal cells are going to make ductal cells. In other words, they maintain their cell-fate. During chronic liver injury, severe liver injury, already in 2011, the group of Klaus Kessner, but also recently the group of Stuart Forbes, they formally proved that there is another way which at the cellular level the tissue can regenerate, and this is by acquisition of high-potent state. Basically, ductal cells can activate a proliferative program, but also a progenitor-like state whereby now they not only make themselves, but they can also make the other epithelial cell type, the hepatocyte. And the same is true, has been shown for hepatocytes. They're able to make kind of cholangiocyte-like cells. And we think this is very relevant because this only happens when you have chronic liver disease in the mouse. When you induce senescence to the hepatocytes, you can see that very well. And actually, this is how patients come to the clinic with a senescent liver, with fibrosis, cirrhosis, or cancer. You have a huge amount of ductal reaction. And of course, we don't know whether in humans this bipotency occurs. But we know that at least in vitro, we can recapitulate that. And this is what I'm going to be focusing on the rest of the talk. So having this in front, whereby now a cell is able to acquire a new fate, this is actually the question we are most interested in my lab. How cells can change their fate and acquire a new state and a new fate. And everything started from the observation that actually this observation actually is from 2010, but it was only published in 2013, that if you damaged a mouse liver, that we had the chance to be able to identify the population that activates proliferation, one of the populations that activates proliferation. So we could isolate these cells and put them in culture. And we saw that actually one single cell could start dividing and make what we start calling them liver organoids. And you can now, this structure that now is formed by hundreds of cells, you can split it, you can mechanically break it into pieces, and each piece is going to make a new structure. And the only thing we do is we just take these cells, put them in a matrix of extracellular matrix, and give them Wnt, HGF, and FGF. And basically, they do the rest by themselves. We don't genetically manipulate them yet. We have a primary culture that can expand ex vivo. The most interesting thing of that is that you don't need to start from a damaged liver. Of course, in a damaged liver, you have already activated the program of proliferation. But in a healthy liver, all the liver health is ductal cells. Now, we know the ductal cell epithelium is the one that's going to start these cultures. Hence, they are called cholangiocyte-derived organoids. We know that the ductal cell that is going to be in a quiescent state, if any of you have for sure have done at least once in your life a K67 staining on a liver tissue, you can see that indeed you have hard time to find even a single K67 positive cell. If you put these quiescent cells in culture from a healthy liver, and you give exactly the same media composition, the same extracellular matrix, they activate what we back then thought they are activating a regenerative program. And indeed, they start proliferating. They make these three-dimensional structures that we cannot distinguish if they come from a healthy liver or if they come from a damaged liver. And they retain this capacity, even if you start from one cell, to make two cell types, the hepatocytes and the ductal cells. Hepatocytes, they will make them if we change the media conditions, but they retain the capacity to change their fate. So very early on, when I started my lab, we had the question, can we use this system to address the question whereby you go from a differentiated state into a bipotent state and now acquire this new fate? Is the organoid system, where we start from a differentiated ductal cell, able to recapitulate that in vitro? We knew it was possible. Is this indeed mimicking what happens during regeneration? And if so, does it allow us to understand which is the mechanism of activating this bipotent state? So that was the work of Luigi Aloia, Michael Mackay in my lab, very talented postdoc and PhD student. Luigi has his own lab at the UCL in London now. And the first question was, yes, we have this observation. So when one ductal cell in culture starts proliferating, can generate two different cell types. But is that indeed mimicking what happens in vivo, or if it's just a correlation? To address that question, what we did is we compared what happens in vivo to what happens in vitro at the transcriptional level. So we isolated ductal cells before inducing any damage at day zero. And then we induced damage, in that case with DDC, which induces ductal reaction. And we collected the cells before, ductal cells specifically, but before they proliferated at day 2. We had quantified that the cells will only proliferate after starting to proliferate at 3 after DDC treatment in our model. So we took the cells before they proliferated, and the peak of proliferation, which was day 5 after the toxin diet. And we profiled them. And in parallel, we did the same experiment. We took the cells before putting them in culture, and at different time points before culturing, after culturing, before the cell divided in culture, which we know it takes 40 hours for a cell to divide in culture. I don't have the data here. I'll be happy to show you the experiments. But yes, we took a lot of time to quantify how long does a given cell move from the G1 state in the cell cycle to enter into S phase. And it's around 40 hours in culture. So we profiled the cells before they enter S phase, before they start dividing, or the cell cycle, and after they generated organoids. And then we compared the in vivo transcriptional profile to the in vitro transcriptional profile. And much to our surprise, what we found is that in vivo, before the cells divide, and in vitro, in the transition from a differentiated to this proliferative state before the cells divide, 70% of the transcriptional changes that occur in vivo, they also occur in vitro during this transition from a differentiated to a proliferative state. And if we look at which are these transcriptional changes, we find that actually there is a lot of epigenetic regulators. More than 55% of known epigenetic regulators, they dynamically change their expression pattern in this transition. And we were very interested on that, and we then digged into some of these genes, and then we were interested on one particular one. I'm just cutting a long story short, which is the TET family of proteins, in particular TET1. And the reason was the following. The reason is because of the function of TET1. So TET proteins is a family of 3 proteins, and in the ductal epithelium, only TET1 is expressed. But it was very well known that TET proteins are essential to induce pluripotency, to induce reprogramming in the pluripotent state. This was already known. And it was also already known that TET proteins, what their function is to, when you have in the DNA, you have the DNA decorated by methylcytosines. So every time you have a cytosine on the DNA, if you put a methyl group, this is a repressive mark. This gene is not going to be expressed. This was very well known. That proteins, they change this methyl group for an hydroxy group. So basically they put an hydroxy on the methyl. And this single molecular change causes a total change on transcriptional. It goes from being a repressive to a permissive mark for transcription. Now all the transcriptional machinery can go there, bind, and express those genes. So of course, getting as a hit TET1 as one of the genes that is highly expressed very early on before the cell divides was kind of telling us that it's going to change the DNA pattern from a repressive to a permissive mark for transcription. So we went on and we asked the question, is indeed TET involved in mediating this differentiated to bipotent state? We took the approach of organoids. We took organoids from a mouse. Then we could deplete TET1 in vitro after generating organoids. And indeed, we could not generate any organoid when we deplete TET1, but they were expanding very well on the parental control lab. And the same is true in vivo during regeneration. So if now you take a mouse model, you induce the liver damage model of Stuart Forbes, where you induce senescence into hepatocytes, and you ask the ductal cells to induce ductal reaction and make not only ductal cells, but also hepatocytes by doing a DDC damage model on top. Then we could see that in a control, this is a PROM1 CRE, rosa ZS green, so every single ductal cell is marked on green, and if it has a progeny, it's also marked in green. So what we saw is that indeed ductal cells makes beautiful hepatocyte clones. But if you now, at the same time, deplete TET1, actually you don't get hepatocyte clones. You also don't have very big clones of the ductal epithelium, suggesting that indeed these TET proteins are important for the transition to acquire this bipotent state, or even further, to proliferate further on. What is the mechanism? We think there is a massive remodeling of the epigenome of these cells before the cells divide. So basically, what we see is that many genes, more than 3,000 genes, acquire this hydroxymethylation mark in the promoter region, suggesting that these genes are being primed for transcription. Not all of them are transcribed, only a subset of them is transcribed, and basically what we find is that important pathways involved in regeneration, like the YAPIPO pathway, the mTOR pathway, are targets of TET1. So we think on a model whereby in homeostasis, we know TET1 is off in the ductal epithelium, and this is just looking at the ductal epithelium. We know TET1 is off. Then just upon damage, before the cell divides, TET1 peaks and actually goes down very fast. And in this, at the moment it peaks, it starts doing its function, which is to put a lot of hydroxymethyl marks in the genome. In genes important to acquire a regenerative state, and that allows the regenerative program to be activated. And TET1 gets off. We don't know what happens after regeneration. This is something my lab is extensively studying at the moment. But basically, this is, we think, one of the, not the only one, but one of the mechanisms that enables a differentiated cell to go into a dedifferentiation state and start proliferating. So all what I've told you is about the epithelium, in particularly the ductal epithelium. What about the microenvironment? What about the niche? And of course here, we have a lot of environment, not only endothelium or immune cells, but also mesenchymal cells. And mesenchymal cells is the ones I'm going to be talking in a second. And that's been the work of Lucia Cordero in the lab, and Anna Dobach, a very talented PhD student and also postdoc. Anna is still with us and is doing another project at the moment. And Lucia, she is doing a postdoc in France. And that actually came from an observation that Lucia did, whereby what she saw is that these mesenchymal cells in pink are literally embracing the ductal epithelium, which I've kind of marked with this dotted line here. So basically you have a mesenchymal cell here in pink that is kind of literally embracing and touching many of this ductal epithelium. And then Anna started to ask the question, how many mesenchymal cells or how many ductal cells are touched by a mesenchymal cell? And is that important during regeneration? Does this change? And here I'm gonna, it's a complex slide, I'm gonna go in detail into it. So this is the homeostatic state of the liver, and is exactly the picture I showed you. So this is the ductal epithelium here in white, labeled in white, and the mesenchymal cells around. What very early on Anna observed is that when you induce damage, and at the peak of the damage, the ductal epithelium expands, but not the number of mesenchymal cells. And actually, we quantified that here. You can see that in gray, the ductal epithelium has expanded already at day 5 after damage. But the mesenchymal cells, which is actually this one, in green, now with this pink oval or whatever, it only starts expanding after the damage is removed and the tissue starts to regenerate at the recovery phase. You can already start seeing them a lot here in that slide at the recovery phase. What does this mean? Anna started counting how many mesenchymal cells touch a ductal cell, and what she saw is that actually it's almost 100%. 100% of the ductal cells, I think the average is 96, and we think it's because we cannot see the tissue in the volume, and we are missing some of them. We are doing these experiments at the moment, but basically virtually almost all ductal cells are touched by a mesenchymal cell. Portal, we know, is a portafibroblast. And this is an homeostasis, but it drops dramatically during damage because the ductal cells proliferate, but the mesenchymal cells have not proliferated yet. Then during the recovery phase, the mesenchymal cells start proliferating, and then they catch up. And then all the ductal cells are again touched by a mesenchymal cell, and that goes back to the homeostatic state, is basically the same. If we look at the ratio between the two populations, what we see is that in homeostatic ratio, we have a 0,3 to 1 ratio. 0,3 mesenchymal cells per 1 ductal cells. This drops just by order of 2, 0.1 to 1 mesenchymal to ductal ratio, and it's recovered to 0.51 and 0.41 during the recovery phase. So basically, you're changing the dynamics between the crosstalk between the two cells. And we were very puzzled by this observation because it was totally correlating with the proliferative state of the ductal epithelium. But we didn't have the means to study this dynamically. We didn't have the means to manipulate this because our culture system was only made by the ductal epithelium. So Lucia and Anna devised a way to put these mesenchymal cells together with the ductal cells. And this is the in vivo tissue, exactly what I just showed you, these mesenchymal red cells embracing the ductal epithelium now in green. And this is a portal endothelium here, so that we don't have. But then Anna and Lucia, they put this together and they try to recapitulate that structure ex vivo in the culture. And for that, they had to use microfluidics. I will be going into details if you're interested, because Lucia spent a lot of time trying to put these two cells together, and what happened is they segregated in the plate. So very interesting, but we were not recapitulating that structure. And only because we could recapitulate that structure, when we started filming these cultures, we could have an observation which was very interesting. Here we put the two cells together using this microfluidics approach. And we couldn't control whether we were going to have cells together or not together. But when they were together, they were always recapitulating that structure. And what we observed, and then we cultured these things in the absence of any growth factor in the media. And what we observed, and you need to focus on the green arrowhead, is pointing to an organoid here that is close to but not touched by a mesenchymal cell. While the yellow arrowhead is pointing to an organoid here that is touched by a mesenchymal cell in red. And the behavior is totally opposite. When there is no contact, the organoid keeps growing. When there is contact, the organoid collapses. Basically, the organoid that was contacted collapsed. The organoid that was not contacted grew, which was kind of telling us that the paracrine signaling, secreted signaling, but was different effect than the juxtacrine cell-cell contact signaling. So we went on and tried to investigate that. And the first thing was to, okay, let's physically separate the two populations. Let's take these mesenchymal cells, culture them, generate media from these ones. We also did Transwell assay. Happy to discuss if you're interested. When we have the mesenchymal cells and we culture them, we collected the media of these cells, and then we put it into naive ductal cells. And we, in the absence of any growth factor in the medium, we asked the question, do these mesenchymal cells produce any secreted factor that allows growth? And the answer was yes. And of course, when you don't have conditioned media, nothing grows. And we saw that indeed they produce HGF, FGF7, which is an analog of FGF10, which is what we use in the cultures, and a little bit of a respondin. And they can retain like this for several passages in culture. What about the other phenotype when they are in contact? What we saw is that now we had the possibility to start playing with the ratios. And indeed, when you go to a 1-to-1 ratio between the two populations, you basically cannot generate any organoid. But if you go to the homeostatic ratio 0:3 to 1, you make an organoid that looks exactly the one I showed you before, with the mesenchymal cells surrounding around the ductal epithelium, but the ductal epithelium does not grow, does not die. When you go to 0.1-to-1 ratio, which was, if you remember, the ratio where in vivo the tissue is proliferating during the damage, actually the organoids grow in the absence of any growth factor in the media. And even if you dump growth factors in the media, you cannot rescue the phenotype. So basically, the cell-cell contact is dominant over any growth factor cocktail that you can add into the media. And we now know that this absolutely correlates with proliferation. So at 0.1-to-1, you have proliferation exactly the same as was happening in vivo. And at 1-to-1, you totally abolish proliferation, even if you add a lot of growth factors in the media. What is the mechanism of this cell-cell contact inhibition? We think there is several mechanisms that explain that, from mechanics to biochemical signals. And one of the signals we've looked into was into notch signaling. When you inhibit notch signaling in the ductal epithelium, and now you add mesenchymal cells on top, now you can totally rescue the phenotype. And this organoid, at a 1-to-1 ratio, where the control will not grow, the organoid where we had inhibited notch signaling prior to put them together with the mesenchymal cells, now it grows nicely. And we know that the mesenchymal cells now in red, they activate notch signaling. This is a HES1 reporter. So basically, the working model here, there is a correlation. We've not proven that in vivo. We just have a correlation between the in vitro and the in vivo. But in vivo, we do prove that when you have too much of the mesenchymal cells, you just undergo collapse. The ductal organoid cannot grow. Basically, we do have apoptosis of the ductal epithelium. When we have a ratio that is around 0.1, which is kind of the damage ratio, they grow beautifully. When you have the 0.3 ratio, or 3-to-10 ratio, you have this structure that doesn't grow but doesn't die. And in the last minutes of the talk, I'm going to go into how we've been using this system to model human disease, in particularly cancer, and one application, which is an unpublished story of the lab. So already in 2000, at the moment we could start establishing these mouse liver organoids from the cholangiocytes that we didn't know back then, we asked the question, can we do the same? Can we translate that into humans? Can we get a normal, healthy liver tissue and grow it? And it took me 1 year to find out the media composition to transfer from mouse to human. I'll be happy to go into the details. I'll definitely go into the details tomorrow on the organoid workshop. But basically, this was a liver donor, someone that dies and donates the organ, so a liver transplant donor. We got a biopsy of this before being transplanted. That was a very precious, very lucky collaboration with Erasmus Rotterdam Center. And we put it in culture, and after tweaking the media composition and finding that we need to inhibit TGF-beta signaling and we need to activate cyclic IMP, then these ductal organoids started to grow. At that moment, again, we were still calling them liver organoids. This would be, now we know, ductal-derived cholangiocyte organoids that we can grow and expand for many months in culture. We can split them the same as the mouse, break them, and each piece is going to make another organoid. They do the same as the mouse. They retain this capacity to make the other cell type. We can have hepatocyte-like cells in vitro. I don't call them hepatocytes because they are not bona fide hepatocytes. So they acquire some function. And as you can already see, some cells are albumin positive in red, and some cells are not. They, of course, they change their shape from a cuboidal to being polygonal, but they are still not bona fide hepatocytes. But they retain some function. They can produce some albumin, some bile acid. They detoxify from drugs. We're improving the system of differentiation at the moment. They even can eliminate some ammonia, at least similar to what HepRG cells can do. But there is still some room for improvement, and as I said, we are still working on it. But that gave us the opportunity to ask the question, can we model disease, human disease in vitro? And that was the work of Laura Brochet in the lab. when she joined the lab, Laura asked the question, can I take liver cancer cells and put them in culture using the human liver organoid culture system I just showed you? And can I grow liver cancer in a dish? That recapitulates in a patient-specific manner the liver of that patient. And the answer was yes. It's very efficient for cholangiocarcinoma. So I would say that it's more than 60% efficient. Every cholangiocarcinoma we get, we can generate cholangiocarcinoma organoids. For the hepatocellular carcinoma, it's not that efficient. It drops to 20%. And it depends on how much proliferative is the tumor. With tumors that are higher than 5% of the population, of the tumor population is proliferative, then we can generate very beautiful hepatocellular carcinoma organoids. If it's a well-differentiated tumor, we can't. And we are improving the system at the moment. She showed that actually these organoids, they recapitulate the transcriptional profile of the original patient they had derived from. Here we have the organoid profile expression pattern, the tumor they derive from, and you can see there is a beautiful correlation not only between subtypes, HCC versus HCC, but NCC versus CC, but also in a patient-specific manner. And they also express some of the classical markers, so AFP. So you have to think that these cultures are all grown in the same media, yet they retain the transcriptional profile of the original tumor. So when a tumor was AFP positive, the organoids were AFP positive. When the tumor was not, the organoids were not. And the same is true for other cholangiomarkers. When the tumor is keratin 19, the organoid it is, and when the tumor was not, the organoid was not. What I found more interesting is that they can recapitulate the histological architecture of the tissue when we transplant them in vivo in a mouse model. And they retain the capacity to, so what we wanted to ask here is, since we had that model that would retain histological organization, the genetic alterations, and the transcriptional profile, we wanted to know whether we could use them to target drug as a tool for drug screening. So this is only a proof of concept. So here we collaborated with the Sanger Institute, where they had a platform where with a robot they were seeding our organoids and they were putting 30 drugs on top of them. And then we asked the question, which drugs are sensitive, which drugs are resistant? And as we expected, as it happens in the population, some the organoids were sensitive, some organoids in red, and some organoids were resistant in white. And then we chose to go for as a proof of concept in vivo for this drug, which is an ERK inhibitor, because some lines were sensitive and some were resistant. And we injected our organoids into the mice. Then when there was a tumor of around 100 millimeters, we started injecting the drug, and we started to inject, or the vehicle of the drug. And while the vehicle grew and again formed this carcinoma in situ surrounded by all of this stroma, the treated tumors, actually, they undergo necrosis. This is tunnel-positive cells, and we saw a remission of the tumor volume. And really, in the last 5 minutes of the talk, I want to show you how we've been using this technology of these tumor organoids to address one question on the mechanism of liver cancer, we think. In particular, how the mechanism of these two genes, RNF43, GNRF3, you don't need to know about the genes. These are two negative regulators of the Wnt pathway. The Wnt pathway is essential during development, also during carcinogenesis. Many liver cancer patients are mutated in some of the Wnt pathway components, APC, axin, beta-catenin. And it was shown in 2012 and 2015 that these two genes are mutated in patients that have liver cancer. These two genes are involved in the Wnt pathway in a way that when the gene is involved in the regulation of the pathway, and it sequesters what is called the free cell receptor, the receptor for the Wnt ligand. And like this, it switches off the pathway. So basically, that's why they are negative regulators. When they are not there, the pathway is on. But for the pathway to be on, you still need to have Wnt ligand in the surface. And I'll be happy to go into the details. So the question for us was, what is the role of these two genes in liver? Because they were mutated in liver cancer. And we were lucky because my collaborator, Bong-Kyung Koo, had generated these mutant mice when he was a postdoc in the Clevers lab, basically. And we depleted these two genes specifically into the hepatocytes of an adult mouse. And then we ask the question, what happens to these livers? And the first thing we see is in a week, there is liver hyperplasia. These livers actually, the liver to body weight ratio is actually significantly higher. The reason is because there is hepatocyte proliferation. But more interesting to us was that after 3 months of the lesion in the adult, we had inflammation and we had fat accumulation. And this is one of the go terms that came from doing transcriptional analysis, transcriptional profile of these livers, and we had lipid metabolic processes very high, very highly enriched. And this is one of these lipid droplet stainings for oil red. And you can see here, I probably can say that here, these are balloon hepatocytes, classical of the steatohepatitis. Very important, there is no insulin resistance. So that's why I don't call it NASH model. We call it NASH-like steatohepatitis. They have fibrosis, upper damage, and upper damage, they also have regenerative nodules, and they terminate having HCC when you induce liver damage. So where does this organoids come in? So we wanted to have that question. What is the mechanism that is driving this steatohepatitis, this liver cancer phenotype? Is it direct? Is it a direct impact of depleting these two genes in the hepatocytes, in the epithelia, or is secondary to all this remodeling, this fibrosis, this inflammation, this fat accumulation? What we see, we did lipidomic analysis on these mice, and actually there is a total change on the lipid species. There is an increase in the unsaturated lipid species in the mutant 3 and 7 months compared to the wild-type glutamates. And there is also an increase on the zonation at 3 months in particular, where it goes from central to portal vein. I'll be happy to, this is mass spec imaging. I'll be happy to explain you the details, but this is marking central, and then here you can see an expansion from central to portal area. What about the direct? So this was clearly an indirect mechanism. The zonation is totally disrupted with also change on the lipid species. What about the hepatocyte inside? What happens to the cell, to the hepatocyte itself? So we took the advantage of the organoids. Now we could grow hepatocyte organoids from these mouse livers. So we isolated hepatocytes. We put them in culture, and then we depleted the gene. And we looked 1 week later, and what we see is these massive lipid droplets compared to these control parental cultures. We counted, and it's not only the number, but also the size of the lipid droplet is significantly bigger. And you can rescue that phenotype by inhibiting with signaling, suggesting that indeed part of the mechanism that we see on these mice, yes, is due to the change on the zonation, but also due to an intrinsic change, a cell-autonomous change on the hepatocytes. What about in human patients? What happens to these patients? I told you that some of the liver cancer patients are mutated in these two genes, either one, the other, or both. We were very lucky. Jessica Zugman in France, she had a cohort of patients published in ICCG where she had done whole genome sequencing and transcriptomic sequencing at the same time, and from the same sample. So we looked at those patients that were mutated in RNF43, or ZNRF3, Z, or both, R and Z. And we saw that indeed they actually have signatures of NASH, signatures of fatty acids, signatures of increased biosynthesis of lipids and bile acid. And also these patients show a very poor prognosis. It's very interesting, and this is the reason for that second curve here. This poor prognosis happens only to those patients where they have Z, or in that case, Z mutations, but no other Wnt mutation. Of course, they have other mutations in other genes, but not in the Wnt pathway. When you have mutations in the Wnt pathway, actually there is some sort of a protective mechanism whereby these patients now, they don't show this poor prognosis. Is also this in part cell autonomous? We were again very lucky here. We had liver cancer organoids that I just showed you before. Some of them were mutated in RNF43, and some of them were not. So we compared lipid accumulation in the mutant versus the non-mutant cancer organoids, and we did see lipid droplet accumulation in the mutants compared to the non-mutant ones. And the non-mutants were very similar to the donor ones. So that brought me to the summary slide, where what I've shown you in the first part is that we are very interested on the switch from a differentiated to a proliferative state. And part of this switch is caused by a change, a massive epigenetic reprogramming that in part, not only, but in part is mediated by TET1 and hydroxymethylation. I've also shown you that the number of contacts between the two populations, in particular ductal and mesenchymal cells, is critical. And the number matters. And too much will kill your ductal epithelium, at least in vitro, and too little allows it to proliferate. And at the end, I've also shown you that you can use adult tissue-derived organoids to recapitulate aspects of disease. I've shown you liver cancer, how we use them to do some drug screening. And I've also shown you how we've used mouse hepatocytes to show that lipid droplet accumulation in particular RZ mutants is in part cell autonomous. And with that, I would like to thank all the people in my lab, past and present members. I've mentioned some of them, Laura Brotier. Now she has her own lab. She was the one doing the liver cancer organoid. She has her own lab in Lyon. Luigi, Michael, Gianmarco, and Germán have been working on the RNZ project. On Anna and Lucia have been working on the mesenchymal ductal epithelial project. And I didn't have time to show the work of the others. And I would like to thank my collaborators in Dresden and Cambridge and Van Den. I'll be happy to answer your questions. Thank you so much. We have done that, not for the liver cancer ones. We did it with the stomach back in 2010. Antibiotics don't make them grow. Antibiotics allow the bacteria not to overgrow your culture, yeah. We don't do it regularly in the lab, but definitely should be possible. I don't see the reason why not.
That's right.
That's a great talk and a great question. We know we've not looked. We are looking at the moment in hepatectomy, yeah. I think... yeah, I don't have a preconceived idea what we are gonna see. My gut feeling could be that probably it's very similar, but I don't know because I think I have the feeling. So of course I don't have a proof of that, except that indeed yes, yes, probably have a proof of what I'm gonna say. Let me check the data. Yes. Yeah. So basically, if you realize what happens is actually the ductal epithelium kind of goes higher than what it should be as a homeostatic state. So basically, I think what happens is that the mesenchymal cells are realized. So you make more ductal cells than what you need. That's why ductal reaction. And then the mesenchymal cells are going there to say, no, guys, now we don't need you anymore. Now let's go back to a homeostatic state, and that's why you have this, again, drop here. It's quite likely that in hepatectomy this doesn't happen because the tissue kind of grows all together. So then most likely you will be preserving the populations and everybody's going to be proliferating. But actually, to be fully honest, I don't know, we've not done the experiments. But these experiments are on the pipeline, yeah. Oh, I think they just do exactly the same. So the growth factors are there, but they are produced by the mesenchyme. So basically, and we kind of looked at it. We did look at it a little bit in terms of expression, and I do think they undergo a little bit of this dedifferentiation that we see when we have all the growth factors there. Yeah, if they don't do that, they cannot proliferate, I think. I think it makes total sense. So basically, as I said, I think that the mesenchymal cells, they are trying. So probably what we think happens is that you make more ductal cells, and then the mesenchymal cells come and catch up, and then they kill some of the ductal epithelium. So basically, they kind of act, and they might help on the clearing, but. Now, if that is decompensated, in that particular case, where you have too many, you've gone not to the 510, which is slightly above the homeostasis, but if you go to 710, 10-10, 1-to-1 ratio, then you've totally tipped the balance the other way around, and then you get a lot of mesenchymal cells, and then you might be getting fibrosis. Of course, we don't have the proof of that, but it's a potential explanation. And usually, because I guess it's about cholangiocyte organoids, we would be checking keratin 19, we would be checking EpCAM, but usually by morphology. So we always look for a morphology that looks like this. Now that I have it here, that it looks very much like this. But actually, if you take ductal cells and you put them in culture, in the media that we published in 2013, they always make that. You don't get anything else that is not a ductal-like structure. For cholangiocarcinoma, I guess it doesn't really matter. For HCC, with the media and what we published in the paper with Laura, it has to be an advanced, undifferentiated HCC. And it seems that mixed subtype, it grows very well. We don't know, probably it's kind of progenitor-like, I don't know. But for HCC, it has to be advanced or undifferentiated. Yeah. Now we checked genome-wide. That was bisulfite sequencing done at base resolution, genome-wide. I just showed you a promoter. For hydroxy, we had to do reduced representation, which it means that it's still genome-wide, but not the whole genome. Again, also at base resolution. And actually, there is massive changes everywhere. Gene body, promoter, CPG island, introns. I just showed the promoter because it's already massive, yes.
Thanks for being here, and see you in December in the next liver seminar. Thank you.